Everyone joining, we're going to wait a couple minutes until everybody gets in the room, and then we will get going. All right, thank you everybody for joining us here today for another one of our TCU alumni webinars. We're joined today by Dr. Laura Luque and Dr. Giri Akkaraju from our biology department. We're going to be talking about vaccines and vaccine development today, which is obviously a hot topic. And it's something that we really wanted to learn a lot about. And luckily we have people that can answer a few questions for us. The way this is going to work is I'm going to ask the questions to our panelists. And if you have a question that you would like answered, feel free to put it into the Q&A section of the Zoom functions, and we'll get to as many questions as we can. Start off, Dr. Laura Luque did her undergrad at Florida International University, and you got your doctorate from University of Texas at Dallas. She focused on infectious diseases, global health, and ethical travel. And Dr. Giri Akkaraju got his undergrad from University of Bombay and his doctorate from University of Pittsburgh. He focuses on a lot of things, but the ones that I could pronounce are viral pathogenesis, targeted therapies for cancer, and drug design and testing. And with that, I want to turn it over to y'all and just kind of get us started with talk a little bit about your experiences and research regarding vaccines and vaccine development.
Okay, I guess I get started. So I did my PhD in molecular and cell biology studying proteins, and that experience led me to my postdoctoral fellowship where I worked with a viral protein for a virus called Sendai virus. And this protein was actually a potential target for a vaccine for a disease caused by a virus called respiratory syncytial virus. So my participation in that project was basically characterizing the protein to see if it was a good candidate as a potential vaccine against respiratory syncytial virus. So I'm familiar with characterizing the potential targets and then the process that is required for developing a functional vaccine.
So hi, my name is Giri Akkaraju. So my experiences with viruses have been more from the point of view of the host or the victim, depending upon your point of view. And since 1987, I've been studying the interactions between viruses and the cells that they infect. So viruses, as you may know, are what we call intracellular parasites. And that's the only way they can survive is by entering a host. And so we have a variety of different defense mechanisms that we deploy against these pathogens. And I've been studying those defense mechanisms for a large chunk of my career. As we've learned more about viruses, we've also started thinking about how we can use them for our benefit. And so for my PhD specifically, I worked on using viruses to deliver genes to cure genetic diseases, something called gene therapy or virus-mediated gene therapy. So that's been my background in viruses.
Awesome. Thank you all for starting us off. So we're just going to jump right to it and get to talking about the COVID vaccine. What is a normal timetable for a vaccine development? And did you have any hesitations with the speed at which these vaccines were developed? That's a common refrain that we have heard.
Laura, do you want to do that, or I can?
Well, I mean, traditionally, when you start a vaccine from scratch, it takes roughly 5 to 10 years from the moment that you start studying the virus or studying the pathogen, identifying potential areas that you can use, to developing a vector or developing a way to delivering this vaccine, and then testing that vaccine in multiple trials. So we call clinical trial number 1, number 2, and number 3. And then you then have the vaccine. So it's a long process, usually when you start from absolutely scratch. I must say that that was not particularly the case with the COVID vaccine. The COVID vaccine was based on technology that was already in the process when the pandemic started. So actually, the COVID vaccine, the Pfizer and the Moderna vaccines were used. The vectors were used in for another process, and they were already in clinical trials phase 2. So we had already many years in advance, and all we had to do was switch the protein.
And so there's no uniform answer about how long a virus vaccine takes to develop. So as you guys may be aware, we're in the middle of another virus pandemic, and that's HIV. And we've been aware of HIV for the last 30, 35 years, and we still don't have a vaccine against it. So vaccine development is very virus dependent, and some viruses are easier to develop a vaccine against. And some viruses are extremely hard. And malaria, which is not caused by a virus, also has been pretty resistant to developing a vaccine against. So on the flip side, we develop vaccines against flu every year. So we have to start designing the vaccine after flu season begins, because we basically have to go out, figure out what strains of flu are going to most likely spread through the population, and then develop a vaccine against it. Of course, the technology, again, is not from scratch, because every year we use the same technology to develop a flu vaccine, but we are developing effectively a new vaccine against flu every year within the space of a few months. So the time taken to develop a vaccine is extremely variable. This vaccine, well, several of the vaccines against SARS-CoV-2, the causative agent of COVID-19, are a completely new technology. And that's the mRNA technology, which we can talk about in the next few minutes. I won't get into that right now, but that's the vaccine I got. So the Pfizer vaccine injects a piece of information from the virus into your body, and you develop antibodies against the protein that is made using that information. And so I had no hesitancy with that technology, because I felt on the face of it that the risks of using an mRNA vaccine are lower than the risks of using a viral vaccine, because most vaccines prior to this contained bits and pieces of a virus or actual live virus that was weakened in some form. So this new technology doesn't even use viruses. It just injects the information that is used to make one viral protein that you develop antibodies against. So to me, it was fairly exciting because it's so new, and it's a great technology because it's versatile and can adapt as the virus changes, and because on the face of it, it seemed safer to me than other vaccines that I've received in the past, like the flu vaccine.
Building off of that, can you take us through some of the differences between the available vaccines? So Pfizer, Moderna, and Johnson & Johnson.
So I'll start by talking about Pfizer and Moderna, and then Dr. Luque will talk about the other one. So Pfizer and Moderna use the same technology where they take a piece of the viral genome. So very quickly, what is a virus? A virus is a bunch of information that's wrapped in a package. And the package is a box usually made of protein. And in some viruses, this protein box is surrounded by a fatty envelope, we call it. And the envelope is studded oftentimes with proteins that the virus uses to glom onto the surface of cells. And in the case of SARS-CoV-2, it's the spike protein that people may have heard of. So that's what's on the outer surface of the virus. So inside this box is the information that the virus carries that can basically make new virus particles. So this information, you may have heard of DNA, which is the information used to make people like you and me. The virus carries this information in a very closely related molecule called RNA. And so when the virus attaches to the surface of the cell, it effectively inserts the RNA into the cell, and then it takes over the cell and uses the cell as a virus-making factory. And so it inserts the information, all the information that the virus needs to make a new virus particle is inserted, and the virus starts to reproduce inside the cell, make copies of itself, make copies of the information, package new virus particles, which leave the cell, which leave the host. Let's say you sneeze, virus particles are spreading through the air and infecting the next person. So this technology takes some of that information. So let's say the virus has 10 different pieces of information that it needs to put together to make a complete virus particle. The vaccine takes one of that pieces of information, namely the information required to make one protein, the spike protein. And it wraps that in a little fatty capsule. And so it uses these molecules known as lipids or fatty acids to basically carry that piece of mRNA containing that one gene or that has information to make one protein into the body by injection. So you inject it into the body, and that then enters the cells around the site of your injection. Say you injected in the arm, this piece of information will enter those muscle cells inside your arm, and those cells will actually take this information and start to make that protein. And so it starts to make the spike protein, but there's nothing else of the virus that's in there. So it can only make the spike protein. And with just one viral protein, you can't make a whole virus. But the body sees this spike protein that the cell is making and recognizes it as being a foreign protein. And when the body sees foreign proteins in the body, inside of it, it starts to make antibodies against it. So the presence of foreign proteins activates your immune system, and cells of your immune system, like your B cells, will actually start churning out antibodies against that spike protein. And so the Moderna vaccine and the Pfizer vaccine use this technology of injecting a small bit of viral information into your body to make one viral protein to then activate your immune system to produce antibodies against that protein. And antibodies, of course, like to stick to the proteins that they're designed against. So an antibody against spike protein will stick to the spike protein. And the way this works is if you get an actual infection, so you inhale some virus particles, because you already have these antibodies in your blood, when the virus gets inside the blood, these antibodies will stick to the surface of the virus, which is covered with the spike protein. And it acts as a physical barrier. So the virus is no longer then able to attach to the surface of cells. So it neutralizes the virus. And we call these neutralizing antibodies. And that's what we hope to achieve with vaccinations, is the generation of these neutralizing antibodies. The other vaccines are a slightly different technology, I would say more traditional technology, and I'll let Dr. Luque talk about that.
Yes, so the Johnson & Johnson, as well as the Oxford AstraZeneca vaccines for some of our audience that may have family members outside of the United States and they're using the Oxford AstraZeneca, both Johnson & Johnson and the AstraZeneca vaccine are using a different technology based on what are known as adenoviruses. So basically, in this particular case, what is happening is that you're using a different delivery mechanism. So here you're using a type of virus known as adenoviruses. Adenoviruses are very common viruses. They are the ones that cause your common cold. They cause pink eye. So they're very common in the population, and they tend to cause not very serious illnesses. But the adenoviruses used in vaccine development, they have been removed of any potential of making you sick. So the adenoviruses used in vaccines will not make you sick. They will not replicate in your body. All right, they will not insert in your DNA. They're basically just used as vehicles to then deliver the same information that Dr. Akkaraju talked about of the spike protein. So basically what scientists do is they modify the genome of the adenovirus, and they change the information for the information of the spike protein. The person receives the adenovirus, the adenovirus enters to your cells, and then the information is delivered inside the cell to start producing the spike protein of the SARS-CoV-2 virus. Once again, your cell starts producing the protein, recognized by the different mechanisms that Dr. Akkaraju referred to, and then you start eliciting an immune response. One big difference between the messenger RNA vaccines and the adenovirus vaccines is that the adenovirus tend to be more stable at higher temperatures. So adenovirus vaccines can be stable at 4 degrees, so in your refrigerator temperature, and therefore makes easier for the delivery and the transportation of that vaccination. And that's probably why you will see this type of vaccine used most in low-income countries as opposed to high-income countries.
Thank you. That was great information. I just want to remind everybody that if you have any questions, feel free to throw them into our Q&A and we'll get to as many as we can. What do you think the next age range of vaccines will be for kids? We've just opened it up to, I believe, 12 to 15-year-olds. What do you think the next age range is?
Laura, do you know? Well, there's still so the 12, so it has been 100% effective. The Pfizer has been tested 100% effective in 18 and lower, and between 12 and 18 years of age, to be 100% effective in that age group. I think they already started the clinical trials on children under the age between the ages of 2 and 12. So as soon as that clinical trial is finished, we will probably see babies being included in the vaccination campaign. Probably by the end of the fall time, we will start seeing young babies being vaccinated or possibly vaccinated for SARS.
Great. We've got a question from the field. Have we learned anything new about the Johnson & Johnson or AstraZeneca vaccine reactions that mirror HIT or any effects on young women?
Right. So what we know so far is that both the Johnson & Johnson and the AstraZeneca, when they were put into the population, because we need to remind ourselves that especially the AstraZeneca has been used in most of Europe and most of Latin America. So we have a tremendous numbers now of information. And what they have found was that this only affects women of reproductive age, and they see effects 3 weeks after the first dose. And what they have found is that 1 in 5 million cases, they have this very serious condition of clotting. But as serious as this condition is, it's actually very rare. So it's 1 in 5 million cases, which is why we couldn't see it in the clinical trials. It was not until we had vaccinated a tremendous amount of people that we were able to see this particular situation, making it even more obvious that it's a very rare event. And it only affects women of reproductive age between after the first vaccination.
Great, thank you. Another question we just got in is, how long does it take the body to reach full immunity with each vaccine?
I can take a stab at that. So for the initial vaccines that were rolled out, the Pfizer and the Moderna, they found a very high level of protection with the first dose. So within a few weeks of being injected, less than 3 weeks, you had over 60% protection. And sometimes it's hard to understand. Well, after the second dose, which in the US was given 3 weeks post the first dose, that was higher than 90% protection. So what does that mean? When you're thinking of percent protection, I think of it as if I got hit by the virus 10 times, 9 times out of 10, my body would be able to fight the virus off, which is a pretty high level of protection for any vaccine for that matter. So these are highly effective vaccines that generated protection fairly quickly. And what does that mean? It means that your body makes antibodies against the spike protein, and those antibodies float around in the blood and are able to fight off infection. So the question is now, how long will those antibodies float around in your blood? Usually speaking, the B cells, the cells that make these antibodies, will last for years. But the efficiency with which they respond to a virus goes down with time. So in the first month, 2 months, 3 months, your B cells are very active. If you get infected with the virus, they can fight viral infection off pretty effectively. But a year, 2 years, 3 years later, it'll start to wane. And sometimes you need a booster shot, which will then give them which will essentially goose your B cells and reactivate them and make them more effective at fighting infection over the long term. And we don't yet know that about this particular vaccine, just because it's been out for less than a year. So we're still in the process of figuring out how long this immunity will last. And just for people to understand how they do this, after they give you the vaccine, to test the efficacy of the vaccine, they'll take a sample of your blood, which contains the antibodies, mix it with virus in a lab, and then infect cells with that virus. And so if your antibodies are working as advertised, if you mix them with virus, they will attach to the surface of the virus and will reduce the ability of the virus to infect cells. And you can actually measure this in the way that I described. So at different periods of time after vaccination, they'll take a sample of blood and look for the amount of antibodies and the ability of those antibodies to neutralize infection. And so it's beginning to look like we may need a booster, maybe a year out, but it's still too early to tell how strong the initial immune response is going to be a year out.
So sticking on the topic of antibodies, some patients who have previously had COVID-19 say they already have the antibodies. Why do I need to go get a vaccine? How long are those antibodies with them? Should they still go get the vaccine? What are the benefits?
Yeah, so the important thing to remember about the vaccine is that it will protect you against a variety of different strains of the virus. And we might discuss this later, but the virus, as people probably already know, changes over time. So the longer the virus lives in our population, it will generate variants. And so these variants will actually change the shape of the spike protein. And the shape of the spike protein is what your antibodies are looking to bind to. So let's say you got infected naturally with one strain of the SARS-CoV-2 virus, and then you get infected with a different strain. The antibodies that you generated the first time may not work against the second strain. However, many of the vaccines that are being used have been tested against multiple different strains. And they've been shown to be effective against even the mutants or the variants that have come up over time. As we've seen, there's a variant that showed up in the UK, there's a variant that showed up in South Africa, and now there are variants that showed up in India. And these vaccines are proving to be effective against all three of these. So especially the Pfizer and the Moderna, which they've tested against the Indian variant as well. So that's why it's important to get vaccinated, because if you got infected with one strain of the virus and then you get a different strain, your antibodies may not be able to protect you, but the vaccine would be able to protect you. So it does help to get vaccinated, even if you got infected with the virus.
Okay, our next question, with the mortality rate being so low among children, why vaccinate them? Is there any risk to a vaccination for those younger children?
Right. So that's what we're testing, if there's any risk for the young children, especially those so young as 2 years of age, between the 2 years of 12 years of age. So that is still remains to be seen, the safety of the vaccines in that type of population. However, although one thing that is very important to remember is that although a person might have mild symptoms or even be asymptomatic, they're still capable of transmitting the disease. And that is one of the main effects of vaccination. So what we know is that vaccines are very good at preventing transmission. So even though you might have a mild case or maybe completely asymptomatic, that doesn't mean that you're not transmitting the disease to others. And actually children, although they might not show symptoms, they are transmitting the disease. And this has been actually well documented. So if the vaccine proves safe for small children, then it will be another way of preventing the transmission of the disease.
Thank you. Considering the long and successful history of vaccination, why do you think there's been so much skepticism for this vaccine?
That's a sociological question more than a scientific question, quite honestly. I do feel that vaccine skepticism came up after what turned out to be a fraudulent report in the scientific literature that linked vaccination to autism. And this link was shown to be false. The paper that was published was shown to have data that was fraudulent. And the person who published it actually lost his medical license. But it was kind of latched on by several individuals who were not scientists, but had an emotional connection to this. Famously, there was one person whose son was thought to be autistic, and she blamed it on vaccination. And she started this campaign. She was on CNN talking about the link between the two and using this paper as scientific evidence, quote unquote scientific. It turned out that her son was not really autistic, but very borderline. But since then, there's been this kind of groundswell of skepticism and fear of vaccinations. I just want to point out that nothing is without risk. So I don't want to completely dismiss this notion that vaccination is completely risk-free. That's not true. But I also want to point out that tens of millions of people die of infectious disease every year. And so it's really important that vaccination protects us, to understand that vaccination protects us from infectious disease and can save tens of millions of lives. And as far as the skepticism goes, people use different logic to kind of try and explain why they're skeptic. The most common one I've heard is it's been rolled out in a hurry. There's not enough tests been done for this particular vaccine. We don't know what the long-term effects are. To which I would first apply the scientific principle is that vaccinations have been around since at least 1796, when it was first documented by Edward Jenner. So we have a fair amount of information on how vaccines work, what they do to you and what they don't do to you. And the new technology used in this vaccine has less components in it that are potentially scary to people, and I feel are safer for that reason. And I'm talking about the Pfizer and the Moderna vaccines, which don't contain any virus at all, but just contains a few molecules of RNA and fatty acids or fats. And so just based on the components of the vaccine, I'm comfortable saying that they're fairly safe. And also that it's been given now to hundreds of millions of people. And any adverse effects should have showed up by now. Of course, long-term adverse effects we won't know, but vaccines in general don't have a lot of long-term effects. The effects are usually short-term and show up fairly quickly. And these vaccines have been around for since last, I would say, October. So we're getting up to about 6 to 8 months. And the adverse effects are beginning to be noticed, and we know that they're very rare. So I'm not sure where the skepticism is. Part of it is from the vaccine skepticism with the connection with autism, which is false. And part of it is spread with the notion that this vaccine was put out there too quickly. But I want to counter that by saying that it's been around for a while and hundreds of millions of people have gotten it with very few adverse effects, extremely rare adverse effects. So I don't know if that answers the question, and I can't answer that question. I don't know why people are skeptical. I'm not.
Thank you. This is a slightly off-topic question, but we have an alum that says you're very effective science communicators, and you would like some advice to those who want to get better at communicating dense STEM topics to a non-STEM audience.
Yeah, so I do this actually kind of on a daily basis when I talk to my family. None of them are scientists, and they have a million questions about the disease, the vaccines, and I try to explain it to them. And if I'm successful at explaining it to them, then I know that I can explain it to anyone else as well. I think is keeping it simple. I think is keeping it simple, but keeping it accurate is very important. And try not to bring any emotional or opinions into the process. I think if you keep it neutral, will make everybody more comfortable in the conversation.
I agree. Thank you. Dr. Luque, one of your areas of study is ethical travel. And so I wanted to ask do you have any thoughts on what travel may look like in the near future with vaccinations?
Yeah, so actually that's a big concern of mine because there's a lot of talk about vaccine passports. And vaccine passports are by nature unequitable due to the fact that vaccination has not been equally distributed, not only within the United States, but also around the world. So asking people to show vaccination will actually be detrimental to a large group of individuals who right now have no access to vaccines, so they cannot then be moving around. But also, you can say, well, okay, they don't have access to vaccines, but then a COVID test. But COVID tests are not for free. You have to pay for COVID tests as well. So, so it's going to be a significant challenge, and it's going to be so. If among the many things this virus has unmasked is part of it is the disproportionate the disparities among health and different groups. So once again, travel will be another area where this particular virus will be unmasking all the inequities around the world. So right now it will be those who have access to vaccines and those who have access to paying the COVID test that will be able to travel, and the rest will lose those opportunities until we find a way to make the vaccine distribution equitable and a test that is easier and maybe even free for everyone.
All right, I've got a couple side effect based questions for the vaccine. We have one alum that is asking about reports of myocarditis in teens and young adults. Would young athletes or distance runners be more or less affected? And then another alum is asking if there's a relationship between vaccines and miscarriages potentially.
Yeah, so the myocarditis is a recent association right now because we don't know for sure if it has any correlation with the vaccine. So right now, as they're starting vaccinated children between the ages of 12 and 18, they have seen these cases of myocarditis, but there has not been a direct link with vaccines just yet. So sometimes what happens is that as you start vaccinating a population, you're also monitoring their health alongside. And sometimes that unmasks conditions that were already there and they're absolutely not related with the vaccine at all. And it's part of the process to filter whether or not these conditions were there before and simply they were not monitored until now, or if there's a true connection between those conditions and the vaccine. And that's where we are right now. So we don't know right now if there's a true connection between specifically myocarditis and the vaccine just yet. We don't know that for sure.
And I don't know if I saw a question about miscarriages and the virus, and infection with the virus can cause miscarriage in women if it infects the placenta, for example. So it has to be a fairly widely spread or disseminated infection for that to happen. And with the related virus, MERS, the Middle Eastern Respiratory Syndrome virus, which was also the same family as this virus, there was a pretty high rate of miscarriages in pregnant women if they got infected with the virus, which to me makes a more effective case for protecting yourself against the virus with vaccination.
Okay, we just got a question. The Pfizer and Moderna vaccines require 2 doses. What is the reasoning behind this? Why is it that one a 2-dose one?
Laura, go ahead.
No, go ahead, Giri.
So it's not uncommon for viruses to require a booster dose. So for viruses like hepatitis B, for example, you need 2 or 3 doses of the vaccine before you attain full immunity. And so generally when you get vaccinated, your body sees the foreign protein, or the foreign object, say virus that you've injected, or part of the virus that you've injected, and this activates your B cells and other cells as well. But B cells are the ones that make the antibodies. And so you can actually count the number of B cells in your blood that are producing an antibody against a particular virus, the vaccine. And you see that it bumps it up with the first dose. And in some cases, if a second dose would bump it up even higher. And so the number of B cells that are producing antibodies against that particular vaccine can be increased with multiple doses. Secondly, as I mentioned, once you bump it up, you get an increased level of B cells producing antibodies, but over the years, those B cells, or the number of B cells, will slowly dwindle. And so by boosting the immunity, that reduces the rate at which you lose those B cells that are producing antibodies against that particular vaccine. And so, which is why in the case of Pfizer and Moderna, the initial vaccination generated about 60% immunity or protection. The second dose increased it up to over 90%, which is why they settled on a 2-dose regimen for that particular vaccine.
Okay, we have a question about herd immunity. What does it take to achieve it? And does incomplete vaccination risk a more virulent strain occurring?
Yeah, so herd immunity will vary from virus to virus and from pathogen to pathogen. So how many people will need to be vaccinated in order to protect those that cannot be vaccinated? That's herd immunity. Will depend on, will vary from virus to virus. And for the SARS-CoV-2 virus, it is estimated that we need to vaccinate roughly 70 to 80% of the population to achieve the herd immunity, where the populations that for whatever reason cannot get vaccinated will be protected by those around them that have been vaccinated. So right now, the United States as a country is at 50%. So we just reached that yesterday. So 50% of the population has received both their vaccines in the United States. That's not the case for the state of Texas. We're still at 34% in the state of Texas that have fully been vaccinated. So once for this virus, it is estimated that we need to reach 70 to 80% vaccination rate.
And I just want to highlight the importance of vaccinating as many people as possible. And what happens when you don't have an immunity against the virus is that the virus will continue to spread through the population. As I mentioned earlier, as it spreads through the population, it mutates and it generates these variants. And the variants actually, some variants are actually better at infecting people. So the current strain in India, for example, has developed a couple of mutations in the spike protein that makes it better able to attach to the cell that it wants to infect. And so the longer time we give the virus to spread in the population, the more mutants are likely to appear. And getting immunized against the virus then basically denies the virus entry into your body and prevents it from developing those mutations. So it is extremely important that we understand that the only way we can contain this virus is by preventing infections. And the best way to prevent infections that we have right now is through immunization. And so not just herd immunity, but even the development of more virulent strains of the virus can be prevented by vaccinating as many people as we can. And that is the bigger danger, in my opinion, about not getting vaccinated, is that you're increasing the chance of viruses that can bypass the initial immune response that you've generated using the vaccines because of the rate at which this virus mutates.
Thank you. We've got a question. One of our alums would like to know more about CRISPR technology and its use of viruses in gene therapy.
That's kind of off topic, but I'll try and answer that question and maybe talk about an application. So the CRISPR technology, more appropriately known as CRISPR-Cas9, is a system for modifying DNA. And so why would we want to do that? So many genetic diseases are caused by mutations in DNA. And these mutations give rise to abnormal proteins that don't do the job they're supposed to do and can harm the patient. A classic example is cystic fibrosis, which is a fairly common disease that's caused by a mutation in a protein that regulates the amount of fluid that accumulates in your lungs. And so if you have this mutation, then fluid can accumulate in your lungs, then making you unable to breathe and making you prone to infections and long-term consequences. So cystic fibrosis is caused by a mutation in a protein that is encoded for in our DNA. And so one way to fix the disease, if we have the ability, was to go in there and fix the mutation. So a mutation in DNA, if you remember your basic high school biology, DNA is made up of bases, A, T, G, C, and the sequence of these A's and T's and G's and C's determine the sequence of a gene, which determines what kind of protein you're going to make from it. So mutation is a change in this sequence, an abnormal change that can damage the protein that's produced from it. So if I could go back and change that mutation back to its original form, I could actually prevent that disease from happening. And CRISPR-Cas9 can do that. CRISPR-Cas9 is actually an enzyme produced by bacteria and used by bacteria to make modifications in DNA. This has been adapted for use in humans, for example. And you can go into a cell, and this has been done in the lab, and you can change a single base, one of the letters in the DNA, thus changing the sequence of a gene, thus changing the protein that's made from the gene. And so CRISPR-Cas9 has been extremely exciting, but it's still fairly complicated and hard to do. And we can talk about that in a different alumni event, because that's a whole separate topic in itself, but which is very interesting. But yeah, I don't know if that answered the question. But
Thank you so much. That was very helpful. So we have an alum that's asking a question about trying to convince family members to get the vaccine. Her mother refuses to get the vaccine due to the use of fetal cells in the testing. Any advice for convincing someone to get vaccination then may have a moral hesitancy?
Right. So actually, the Moderna and the Pfizer do not use fetal cells in their production. It's only the Johnson & Johnson and the AstraZeneca that do. So if that's an objection, they can, if possible, especially now in the United States, where you can actually find, you're in a position now to find the vaccine that you want, which was not the case before. Technically speaking, you could go and try to find Pfizer and Moderna, which bypass that particular obstacle if the person feels morally and through their principles that is an objection to fetal cells, so they don't receive the Johnson & Johnson. They can receive the Pfizer and the Moderna, which don't use fetal cells.
And that's a difficult question to answer, honestly, because you have to follow your conscience as far as that goes. But I worked on a project a few years ago where we worked on studying a virus known as hepatitis C virus. This virus infects 120 million people a year, and it was effectively a death sentence. And today we have a drug that works really well against hepatitis C, which makes it a manageable disease. And the drug was developed out of research that was done on fetal cells. And so I feel it's a cost to benefit as well, is that hundreds of millions of people's lives have been saved because of work that was done in those cells. So, I mean, that's one calculation that I use, for example, in determining whether a particular cell type should be used in research or not.
Thank you. We've got time for about one more question, then we can start wrapping up. Are there any signs of vertical transmission of COVID antibodies to infants?
So mother to child, we don't know a lot about what is going on between COVID with pregnant females that receive the vaccine and how that affects their offspring. So we have very little information about any vertical transmission, so mother to child. So if the mother has the vaccine, will the antibodies be transmitted to the child? Probably not during the pregnancy, but actually, if after birth, during breastfeeding, then it will be transmitted that way. So that's one of the most important reasons of breastfeeding, is because you do transmit your antibodies in that process and protect your child, especially during the first 2 years of age, when they're so vulnerable to diseases. But as far as we can tell, we don't have information yet if, as the mother is pregnant, will the antibodies cross the placenta. We don't know that for sure.
Great. With that, do y'all have any closing thoughts for our alums?
Yeah, I see a question about the logic behind CDC relaxing masking requirements. And I would say that I would not take my mask off if I'm not sure that everybody around me was vaccinated. I still think that masks can protect you from getting infected. Avoiding crowded places can protect you from getting infected. Until this virus has been completely eradicated, we all should be fairly cautious. The CDC may have relaxed the masking requirements partly because of fatigue. I think if they hadn't done it, people were starting to do it anyway. But if you're making a personal decision about wearing a mask, I would say, unless you know that people around you have been vaccinated, you should wear a mask and avoid crowded places. And if you do go to crowded places, please wear a mask. It will keep you safe.
Yeah, and just, well, thank you very much for attending this seminar, first of all. I think that is great, and disseminate the information, hopefully, that you found useful here. But also, I think we have many lessons to learn from other countries as they are going through this process. So the United States has done tremendous efforts and success in lowering the rate of infection. But as Dr. Akkaraju says, we're not there yet. We're almost there, but not quite. So we still have to wear a mask and hopefully get vaccinated. Because when that doesn't happen, we can have a situation like what we're seeing in India, which is very scary and very, very dangerous. So hang in there. We're almost there. We just need to wear a mask a little bit longer, and then we'll hopefully be over this hump.
All right. Thank you so much. With that, I'll close this out. Thank you everyone so much for joining us here today. If you haven't attended an event with us in a while, please go to alumni.tcu.edu and update your information. So that way you're getting accurate emails for your area. And with that, get vaccinated and go Frogs.
Thank you for inviting me.
Thank you for inviting me too.